Okay, everyone, if you can hear me, write the number 5 in the chat. Write the number 5 in the chat if you can hear me. Write the number 5 in the chat. Good, good, good. Excellent. Okay, no need to write 5 anymore. Good job. Okay, so we got some feedback since the last lecture, and 3 hours of questions was a little bit too much. So people requested that I do a little bit of lecturing, a little bit of question answering, and a little bit of advanced material. Some people said the material was too easy.
Hey, John.
Yes?
Hey, John, you should mute your music.
My music is off.
Yeah.
My music is off. Are you still hearing it?
I can hear it. I can hear the music.
My music is completely off. Are other people hearing
maybe other people.
Are other people hearing music? If you're hearing music in the chat, write the number 7. Most people are saying no. I don't have any music on. It's completely silent in my office. Do you still hear something, Shun?
The music stopped.
Okay. Can you hear me okay?
Yes.
Okay. So some students said that the material was too easy. And so let me just say a few things. As the class progresses, it will get harder. The material will get more advanced. But if there is a range of ability, some students are starting off here at level 1, and some students are at level 10. So I have to try to make sure that everyone can learn the material. So I have to try to take the people that are at level 1 and raise their understanding up. So this is why we start the class talking about a little bit of biochemistry and talking about fundamental experiments, okay? Because we need to start very basic, and then it will become more advanced, okay? But for today, I thought I would lecture on the hardest topic in the world. The hardest topic in biology in the world, which is how did life evolve? And I thought this was a good topic to start because lots of questions that people were asking were kind of related to this. And the lecture about eukaryotes versus prokaryotes has a lot of evolutionary themes, and there's a lot of evolution to discuss in this context as well. So I thought today that I would start the class by lecturing on the most hardest topic in the world, which is how did life evolve? And then nobody can say that the material is too easy. Okay. So imagine what has to happen for the first organism to evolve. And we have a name for this organism. We call this organism LUCA, which stands for the last universal common ancestor. Okay? So the first organism that then gave rise to all the other organisms, which then gave rise to the tree of life. This we call LUCA, the last universal common ancestor. Okay, so think about what has to happen for this organism to evolve. Let's talk about what it needs. Okay, so one, it needs a container. It needs some kind of a lipid bilayer, a membrane. Okay? What else does it need? It needs the basic building blocks of life. So in theory, that would either be DNTPs or possibly RNTPs. And it needs amino acids. Okay? And it somehow also has to know how to replicate. Okay? It needs all these things, and the reason why this question is so hard is because it needs all these things simultaneously. That means it needs them all instantly, at the same time. Without just one of these, if you just had 2 and 3, you couldn't get life because molecules might replicate, but then they would diffuse. Okay? So if you don't have a container, you don't have life. And if you just have a container and reagents, but you don't know how to replicate, you don't have life either. So the reason that this is so hard to evolve is because you need all these things simultaneously. Okay? So let's talk about hypotheses about how this happened. One second, I just need to shut off my notifications. Okay. So one hypothesis is called, I'll type this one out. One hypothesis is called RNA-world. Have you guys ever heard of this? If you have heard of this, in the chat, write yes. Only one person has written yes. So it seems like you guys have not heard of this yet. Okay, so let's talk about this. The RNA-world hypothesis is a hypothesis that in theory should explain how we can derive at least these two things at the same time: building blocks and a knowledge to replicate. So RNA can, in some cases, be an enzyme. So what does that mean? That means RNA, type this out, RNA can have catalytic and metabolic activity. Okay? So there are some RNA enzymes. Let's say there was a RNA enzyme. Maybe it looks like this. Okay? Because RNA is single-stranded, but sometimes it can form what would be called ultra structures. And these would be things like hairpins. So this would be a hairpin. Okay? These structures can sometimes come together to form an enzyme. That means RNA can evolve in a way where you can get a site that might do something. Okay? So that's the first sort of premise. Let me write the premise of RNA-world hypothesis is that RNA is a unique molecule because it can self-replicate and it encodes its own template. Okay? So the RNA world is this idea that there was originally some RNA molecule. And this RNA molecule could take pieces of RNTPs, and it could synthesize these by looking at itself or potentially its sisters, and it could make new copies of itself. Okay? So the RNA world says maybe the first self-replicator was an RNA molecule that can replicate itself. Okay? And this is the most favored hypothesis that leads to the evolution of the last universal common ancestor. The RNA world is sort of like our best hypothesis. Okay? But it has lots of problems. Okay? So we're talking about a topic that no one understands. No one knows the answer to this question. Oh, I'm gonna mute somebody here. Rao, Seed, Raymond, please mute. Got it. Okay. So let's talk about some of the problems with the RNA world hypothesis. Okay? If it was true, we should be able to replicate the first organism in a test tube. If RNA world hypothesis was true, you should be able to take a tube and you should be able to add RNTPs into that tube, and maybe you have to test many different conditions. Okay, maybe you have to do a giant screen. Maybe there's even greater than 1,000 different conditions that you might test. But if it was true, you should just be able to add RNTPs into a tube, and eventually, in theory, you should assemble the self-replicator. Okay? People have tried that, and it doesn't work. Okay? So that's evidence against RNA world. Okay? There's also a question about is it even possible to take randomized RNTPs and randomly assemble them into an enzyme? Is that even possible? Like, we can actually mathematically calculate this. So these self-replicating molecules, the only self-replicating molecules that we have been able to produce in the lab are about 100 base pairs long at least. So they're all greater than or equal to 100 base pairs. So the idea that you could take random RNTPs and randomly assemble them into a self-replicating molecule that matches one of these ones that we know that can replicate, the odds of that are 1 out of 4, because there's A, U, G, and C. 4 to the 100th power. Okay? Let's calculate that. Hang on. My computer's going slow. I calculated it earlier. It's, oh, here we go. Oh. It's a number times 10 to the 60th power. Okay? It's gigantic. So the odds of that happening are extremely rare. Okay? So this is evidence against the RNA world hypothesis. Okay? But there's more evidence against this. Let's talk about more. Okay. In order for RNA-world to be correct, you need to have a container. Okay, I talked about this. If we can slide up. The things you need for life to evolve, you need a container. Okay, we talked about this. And in theory, in that container, you could collect. Please mute your microphone. Hang on, I gotta mute. Somebody is driving. Zao is driving. You should not be driving and watching this lecture. Okay. In order for this to happen, you'd need a container. Okay, so you'd need a lipid bilayer to collect the, not DNTPs, to collect the RNTPs. Okay. And in theory, then, in that container, you could evolve a self-replicating molecule. But there's a problem, okay? Lipid bilayers, zoom in, lipid bilayers are not permeable to RNTPs. Let me see if I can, I don't know the spell. I don't know the spelling. That's okay. What that means is, if you have a lipid bilayer, there's no way for the RNTPs to get in. There's no way. They can't just go through. And if this is the first organism, we haven't evolved. So let me write this out. If this is the first organism, we have not yet evolved porins or receptors or transporters. So how can the first container concentrate the RNTPs? Nobody knows the answer to that question. Nobody knows, okay? So this is more evidence against RNA-world hypothesis. Okay? There's more. Okay? There's more evidence against this. So there is a professor at Harvard. His name is Jack, spell his name right, Szostak. Okay? He's at Harvard. He studies this at Harvard. He won a Nobel Prize for studying telomeres. Okay? So he's a very, very smart guy. He has studied these quote-unquote self-replicating RNA molecules, which people think, I can't type, precede the first organism. Okay, he studied these. And when you look at the catalytic rates, okay, the catalytic rates of these enzymes, so let's talk about that. The catalytic rate, that means how fast can it synthesize copies of itself. When you look at the rates, the best self-replicators, which are, keep in mind, they're 100 molecules. They're 100 base pairs long, at least. The best rates are, they can ligate 14 base pairs per 24 hours. Do you see how slow? You see how slow that is? If you take, this is good because yesterday, or the first day, we talked about polymerases. Compare this to a polymerase, which writes DNA, or in some cases RNA. Tac, or let's do a high fidelity one. So fusion, which is a high fidelity polymerase, that can write every 15 seconds 1,500 base pairs. Okay, this thing ligates only 14 base pairs in 24 hours. That's so slow. That's so slow that the molecule falls apart faster than it can replicate itself. So the only way you get reactions of these self-replicating molecules is if you have many of them and you fill a tube with many of these, and then they can collaborate together and make maybe one copy before they all fell apart. Do you see how this will never work? This will never lead to the evolution of LUCA, because the molecules will fall apart. Okay? So this is more evidence against RNA-world. Okay? There's more. Okay? In this work, studying these molecules, the molecule itself is not enough. It needs a cation called magnesium. So these enzymes can't function unless they have a magnesium cation. So these enzymes can't function. I'm going to mute somebody. Hang on. Okay. So that adds another piece to the puzzle, that if RNA-world was true, you also need a source of magnesium. I'm not sure if you're going to have that all at the same time, all in the same container. And remember, it's questionable if the magnesium can pass through a container. Okay? So this is all evidence against RNA-world. Let's keep going. There's more. There's more evidence against RNA-world. Okay? If you take one of these self-replicating molecules, okay, and you mix it in a tube with different RNAs, let's make them different colors. Let's say a green RNA, a blue RNA, a rainbow RNA. One big problem is this molecule, this self-replicator, if it was to replicate, it would have to be able to see, okay, here's an eye, it would have to be able to see this one, its copy, in a group of a mixture, and they cannot tell, they can't see that. So let me say this in a different way, a better way. The self-replicating RNA molecules cannot distinguish between themselves and other parasitic RNAs. So what happens? If you mix this in a tube, what happens is this replicates this one, and it replicates this one, and it replicates this one. But these ones can't self-replicate. They're not the right code. So there's no ability to distinguish between itself and parasitic messenger RNAs. This is a tough point to convey. If you're understanding this point, type 3 in the chat. If I should keep explaining this, if I should try to explain this again, you're understanding it. Somebody says they're understanding it. Okay, they understand. Okay, good. Very good. Okay, excellent. Okay, very good. Okay, there's more problems. Let's keep going. There's more problems with the RNA world hypothesis, okay? There is a problem called the Enantiomer problem, okay? And you might have heard about this a little bit in biochemistry when you hear things like racemic mixture, okay? Now let's talk about this. We each have a left hand and a right hand, okay? And these are different. These are actually different structures. They're not the same. They're mirror images, okay? And it's the same for RNTPs, DNTPs, and amino acids. There are both left-handed and what we call D versions of these molecules. And life only uses L. So in the case of amino acids, life only uses L amino acids, okay? So not only do you need all those 4 things for the RNA world to work, you also need a way for this to work, for RNA world hypothesis to work. You also need a mechanism to purify L, let's call them left-handed molecules. You need to separate out the D versions or the D variants, okay? And there's a problem because when inorganic chemists synthesize RNTPs in reactions which they think are close to the early conditions of the Earth, those reactions form racemic mixtures. And racemic mixtures are, you have equal amount of the left-handed and the right-handed. And that's a problem because life cannot evolve if there's an equal mixture of these 2 variants. Life cannot evolve if that happens. It's got to take one and get rid of it. Okay? So do you see how this is more evidence against RNA world? Because it could only happen in a universe with just L variant RNTPs. Okay? Okay. So, despite all that I just told you, RNA world is still our best hypothesis. Okay? That's pretty bad. Because it's not really a good hypothesis. There are so many problems with that hypothesis. I wish I had the answer. But I don't. So do you see how hard this problem is? Okay, so let's think about other hypotheses. What other hypotheses are there? Okay, so let's move on from RNA world. Let's move on. Hypothesis 2. So this is hypothesis 2. Is called metabolism first. This means that small proteins in enzymatic pathways capable of producing energy came first. That was the first step in evolving the first organism. This is the metabolism first hypothesis. Okay? Now this also has huge problems. This also has major problems. Because if any of you know anything about biochemistry, right? When we look at like the Krebs cycle, okay, or like respiration, there's always like one enzyme, I'll write this out, one enzyme, two enzyme, three enzyme, 4, 5, and they're all in a pathway to get an end product Y. And if you don't have 1, 2, 3, 4, and 5, it doesn't work. So this also has a major problem, because then in the metabolism first pathway of evolving life, not only do you need a container, not only do you need amino acids, not only do you need that, but you also need to evolve. You need to evolve at least 5 enzymes all simultaneously. Otherwise, it could never reproduce itself. It could never create a chain reaction unless you have all these things at the same time. Okay? So this hypothesis is also bad. It also has the same problems or similar problems to RNA-world. Okay? Let's talk about another hypothesis. This one's funny. Okay, hypothesis 3. Hypothesis 3 for how life evolved is, and this is a serious hypothesis, people actually posit this, that we were seeded by asteroids or comets. Okay? And this hypothesis is, here's the Earth. Okay? The continents look different. But here's the Earth. And there were comets that bombarded the Earth and struck the Earth. And on these comets were molecules of life. Okay? This is one hypothesis. Now, the only evidence for this, there's 2 pieces of evidence for this. One, there's a rock called ALH84001. Okay? This is a rock. Okay? And it was found, so ALH84001 is a rock that was found in Antarctica, and it's a meteor from Mars. It's a meteor from Mars. So we also, if this is Earth, we have another planet. This is the 3rd planet. This is the 4th planet, Mars. We should all know this. There is a rock that was found on Earth that came from Mars. Okay? So finding this rock was evidence that interplanetary travel happens at least by rocks. Okay? And the thought was that a giant asteroid hit Mars, and when it hit Mars, it exploded, and it sent a rock all the way, and it got caught in Earth's gravity, and it went down. Okay? And on this rock, if you look with a microscope, They claimed there were fossils of microorganisms. And this was published in Science. Okay? Now, this is debated. This is heavily debated, okay? I don't think this is true. This is probably not true, but it was published in Science, okay? So they thought this was evidence. This was evidence for the seeded by asteroids hypothesis of how life started. There's also another piece of evidence for this, okay? It was just recently published. We recently analyzed rocks from a comet. And on the comet were proteins, and all they were glycines. They were just a couple glycines. They look like this, okay? But this was evidence that you can find amino acids on comets. Okay. So that's it. That's the only evidence we have for this hypothesis. So this hypothesis is also bad. Okay. Who knows? Who knows? Okay. So let's keep talking about this, because you wanted to talk about the hard stuff, the hard problems in science. There's another hypothesis. Okay, here's my hypothesis. Because a lot of people ask in the questions, and a lot of people ask, they say things like, can you talk about this? And they say things like, what's your opinion on this? Okay, so I'll tell you my hypothesis. Okay, so hypothesis 4. Okay. Imagine you start with a container. A container that moves. Hang on, gotta mute somebody. Okay, imagine you have a container. And by moves, I mean like it floats. And it's stable. Okay? This solves one of the problems. If this was true, it solves one of the problems. One of the problems with all the other hypotheses is all the hypotheses 1 and 2, they rely on the idea that there is like a factory. Okay? So if this is like a factory that produces, I'll write this out, I'll type this out. Hypotheses 1 and 2 rely on the idea that there is a factory that makes RNTPs, or in the case of the second hypothesis, amino acids. Okay? That's pretty unlikely. But what they think is, so if here's the Earth, if here's the ocean, if I could get a blue one. If this is the ocean, you go down deep into the ocean. Deep into the ocean, there are these, what they call hot vents. Okay? And these hot vents are like these rocks. And in these rocks are little holes. Okay? They're porous. Okay? And underneath the surface is lava. Hot lava, which cooks these hot vents, these porous rocks. And they think these hot vents might be like a thermocycler. Okay, you see the thermocyclers in the lab? Thermocycler is a machine, looks like this. You open it up and it's got a little heat block. And it's what you do PCR in. Okay? They think these hot vents are like little thermocyclers. Okay? And they think that in each of these caverns, that solves the container. Okay? That would check the box of the container. These little holes. And that would check the box of the RNTPs, because maybe they're like a factory that synthesizes RNTPs. Okay? This is one idea. But then there's another problem. There's another problem. Okay? The problem after this is then, if this is true, and the first cell was a little hole in a rock that had RNTPs, if that's true, how does the first organism get a lipid bilayer? How does the first organism escape? It's impossible. It's impossible. If you're in these vents, it's impossible to leave. You cannot leave. Because let's draw a picture. Let's go slow. Let's draw a picture. So here's a rock. And in this rock are little pores. And we're heating it. So in these little pores, we're synthesizing molecules. And the temperature is going up. So the degrees Celsius, the temperature is going up and down and up and down and up and down. And we're synthesizing these molecules. And their thought is, at some point, one of these molecules forms a self-replicator. And it's able to replicate itself. And it's able to replicate itself. And it's able to replicate itself. Okay? That's what they think. But if that was true, how can the self-replicator ever leave or exit the rock? It can't, because it doesn't have a lipid bilayer. There are no lipids, okay? So I don't like this. I don't think this hypothesis is true. Maybe. Who knows? Maybe. This is like the best we have. The best hypothesis we have. But it doesn't answer this question of how can it leave? So I think the better idea, my hypothesis, hypothesis 4, is you have this container, a lipid bilayer. You start with a bilayer. Start with a bilayer that's stable. And it somehow develops a collecting ability. Okay? So it floats around, and it somehow is collecting little pieces inside of it. And then maybe from that, you somehow get self-replication. If that was true, then maybe that's how Luca evolved. I don't know. I don't know. This is hard. This is a hard problem. You guys wanted hard problems. You wanted advanced material, okay? Let's go further. Okay, so now this I can use this now to segue into a really good question. So we've kind of gone through these. This is a really good question that relates to this from Mubashar Hussain. He says, there are 500 amino acids in nature. Why do humans only code 20? Which organisms code completely different amino acids from humans? Okay, I'm going to skip this. I'm going to try to answer this. There are 500 in nature. Why do humans only have 20? So, imagine there are 500 amino acids. The first way to answer this question is, were there 500 amino acids in the original container of Luca? So we're talking about the first organism. If you're trying to answer why does life only use 20 amino acids, the answer is because the first organism only used at most 20, probably less. And the reason it's probably less is because what are the odds that the container, I'll type this out. What are the odds that the first container had all 20 or all 500 amino acids? Very low. Probably only had a couple. Okay. So if I'm trying to answer why does life only use 20 amino acids, my answer is because however it evolved, Luca probably only had 20. Okay. The other answer to this, so that's answer 1. Answer 2. The other answer to this is a topic called parsimony. And parsimony means in science, usually the simplest answer is the most true. This is just sort of like a philosophical rule or a heuristic. Okay? And it's not parsimonious for the first organism to evolve with 500 amino acids, because that means there have to be 500 amino acids in the first little rock. Okay? Especially when we can build complex machines with only 20. You don't need 500. You only need 20 at most. Okay? So that is probably in this question I wrote because 20 is sufficient. So that's how it evolved. Parsimony. And then I wrote, when the first organism Luca evolved, there probably were not 500 around. Okay? And now the third piece comes in. tRNA only matches to 20. Okay? So in our cells, in a cell, the way that we make proteins is there's a ribosome. And in the cytosol, there are different tRNAs. There's a red one, black one, a blue one. And these tRNAs match codons, and they bind to amino acids. Each of these tRNAs, each tRNA has a gene corresponding to it. So if Luca used 500 amino acids, it would also need 500 tRNAs. That's impossible. Do you see? That's impossible to evolve, because that would mean you would need 500 things simultaneously to evolve life. That's impossible. Okay? So that's a really good question. Hard to answer. I tried my best. I'm going to skip these. Now let's go to a bunch of questions on... Oh, let's take a break. This is a good time to take a break. So it's 7:50. We'll take a 10-minute break. We'll come back in 10 minutes. Alexa, set a timer for 10 minutes.
10 minutes, starting now.